Hello students, I'm C. Pavani, Assistant Professor at BVRIT Narsapur and from Pharmaceutical Engineering Department, and in today's video we are going to learn about biosynthesis of fatty acids. So biosynthesis of fatty acids, before going to the biosynthesis of fatty acids, what is fatty acid and how it is synthesized in the body? Let us have a brief of it. For example, whatever the excess food you are taking, generally whatever the food you are taking, it gets converted into glucose. The excess glucose which is not utilized as energy is stored in the form of fatty acids. So the conversion of that glucose into fatty acid is nothing but biosynthesis of fatty acid. This happens in the cytosol of cell, which is a primary site for this process. As we discussed in the previous video, in mitochondria you can see beta oxidation of fatty acid, and in the nucleus you can see, not in the nucleus, in the smooth endoplasmic reticulum you can see omega oxidation of fatty acid. In the cytosol or cytoplasm, you can see fatty acid synthesis. So remember these regions where what happens? Beta oxidation happens in mitochondria, smooth endoplasmic reticulum omega oxidation, and fatty acid synthesis in cytosol. So you can see the cytosol in many cells like liver, mammary gland, kidney, brain, etc. And for this process, for the biosynthesis of fatty acids, lot of enzymes are involved in this process. Along with enzymes, it also requires ATP, biotin, NADPH, HCO3, bicarbonates for the source of carbon. And the immediate or the initial molecule of this process is acetyl coenzyme A, and the end product of this process is palmitate, which is a 16-carbon fatty acid. So most of the acetyl coenzyme A which is needed for this process is synthesized or derived from the glycolysis pathway of carbohydrates. Coming to steps involved in this biosynthesis of fatty acids, there are 5 steps are involved in this. First one is citric acid shuttle, followed by production of acetyl coenzyme A, production of malonyl coenzyme A, and the third step is fatty acid synthetase complex, and fourth step is steps involved in fatty acid synthesis. Last step is palmitate acid synthesis. So let's get into one by one. Step 1 and step 2 we will discuss together. So first, acetyl coenzyme A this is the end product of many processes. As we discussed, acetyl coenzyme A is the end product of beta oxidation procedure, omega oxidation, as well as this is also synthesized from the glycolysis cycle. So there is lot of acetyl coenzyme A in the cells which gets converted into citrate. Acetyl coenzyme A cannot itself enter into cytosol. This is mitochondria of the cell. So acetyl coenzyme A cannot enter from mitochondria to the cytosol directly. So it has to convert into one of the soluble form, that is citrate, and from citrate can easily enter from mitochondria to the cytosol. Okay. In the cytosol, citrate is a 6-carbon compound. In the cytosol, it gets converted into 2-carbon acetyl coenzyme A and 4-carbon oxaloacetic acid. This oxaloacetic acid again enters into mitochondria for the Krebs cycle or citric acid cycle, for the progress of Krebs cycle or citric acid cycle. Krebs cycle is very important for many of the biological processes, as each and every product of Krebs cycle is utilized in the synthesis, or it is a precursor in the synthesis of many amino acids in the body. So this 4-carbon oxaloacetic acid enters back into the mitochondria, and acetyl coenzyme A, which is utilized for the further process of this fatty acid synthesis. So in the second step, acetyl coenzyme A gets converted into 3-carbon malonyl coenzyme A in the presence of ATP, biotin as well as carbon dioxide. ATP is converted into ADP by utilizing one of the phosphate in this process. And this whole process occurs in the presence of enzyme acetyl coenzyme A carboxylase. And this bioacid or fatty acid synthesis happens only up to middle chain fatty acids, up to 14 carbon chain and 16 carbon chain. More than that happens outside the cell. In this, the next step, after the malonyl coenzyme A is processed, then the next step is fatty acid synthase complex. The name itself suggests in this fatty acid synthase, there are 7 enzymes involved in this. So all these 7 enzymes together is called as fatty acid synthase complex. This multi-enzyme polypeptide complex also contains acyl carrier protein in its structure. Along with that, it also has 4-phosphopanthine, or this is a form of vitamin pantothenic acid. We also call it as vitamin B5. This is also one of the synthetase enzyme. This is also involved in the many fatty acid synthesis chains. And this, you can see this complex which has 7 enzymes, 2, 3, 4, 5, 6 and 7 enzymes. And ACP is one of them. As we discussed, acyl carrier protein is one of them. Along with the 7 enzymes, it also has vitamin B5, a form of vitamin B5, that is 4-phosphopanthine. So it has 2 monomers. You can see one monomer here and the other monomer. So this is called as dimer. The next step is fatty acid synthesis. By utilizing fatty acid synthetase complex enzymes, this whole process happens, that is fatty acid synthesis, and there are 5 steps involved in this. First step is condensation, second step is reduction, and third step is dehydrogenation. Fourth step is reduction again, and a repetition of the steps several times depending on the length of carbon chain. For example, let us start with the initial step, glucose. As we discussed, glucose, the end product of glucose is acetyl coenzyme A. Acetyl coenzyme A gets converted into malonyl coenzyme A. This is 2 carbon, and malonyl coenzyme A is 3 carbon, and this requires ATP, carbon dioxide as well as biotin, as we discussed in previous slide. This initial step happens in the presence of acetyl coenzyme A carboxylase. Next, this malonyl coenzyme A gets converted into malonyl ACP. Wherever ACP, it is acyl carrier protein. So it gets converted into malonyl ACP in the presence of malonyl CoA ACP transferase. Next step, the first step of this synthesis process, fatty acid synthesis process, the first step is condensation. This involves 3-ketoacyl ACP synthetase 3 enzyme for the conversion of malonyl ACP to 3-ketobutyryl ACP. So here you can see 3-keto, butyryl is nothing but 4 carbon atoms and ketone group at the 3rd carbon atom. 3-ketobutyryl ACP molecule. This also involves utilization of acetyl coenzyme A in this process. And next, 3-ketobutyryl ACP is reduced into 3-hydroxybutyryl ACP in the presence of 3-ketoacyl ACP reductase, and this happens in the presence of energy source, that is NADPH plus H plus. And this, after this reduction process, it gets like 3-hydroxybutyryl ACP gets converted into trans delta square butyl. See here, in this second step, this gives hydroxy group at the 3rd carbon. If you start counting from here, this will be the 3rd carbon. So third step is dehydrogenation. It is also called as oxidation. In this step, 3-hydroxybutyryl ACP gets converted into trans delta square butynoyl ACP in the presence of 3-hydroxy acyl dehydratase, and this happens by the removal of water as well. And in the fourth step and final step, this is reduction step, where this also, so as you can see, both of this reduction steps, second and fourth, utilizes NADPH plus H plus for the conversion. And for this, in this reduction, you can see the final product as 4-carbon butyryl ACP, and this 4-carbon butyryl ACP is again involved. This is a cycle repetition. You can see this is the fifth step. This is the repetition step. This happens until you get a final end product. For example, if I want a 12-carbon, 14-carbon, 16, the end product of this, whatever we are expecting, is palmitic acid. If you want the end product as myristic acid, no, like we cannot synthesize all this process. It happens inside the body. If the myristic acid is needed inside the body, then this whole process happens 6 times. If the end product is palmitic acid, this whole process happens 7 times. And if the end product is lauric acid, then the whole process happens 5 times. So based on number of cycles, you will get the end product. So the last step is palmitic acid synthesis. So palmitic acid is a 16-carbon chain, and for the from acetyl coenzyme A to the addition of malonyl coenzyme A, it requires total 7 malonyl coenzyme A in the whole process. Along with that, it requires 14 NADPH, because at 2 reduction process we require 2 NADPH for each cycle. So it requires 14 NADPH, and the end products of the 7 cycles is palmitate, as well as you have 7 carbon dioxide molecules, 6 water molecules, 8 coenzyme A molecules, acetyl coenzyme A molecules, and 14 NADP plus molecules. So let us see the overview of this whole cycle. First one is citrate shuttle. We call it as citrate shuttle because acetyl coenzyme A by itself, it cannot enter from mitochondria to the cytosol of the cell. So it has to get converted into citrate. After citrate entered into the cytosol, it gets divided into oxaloacetic acid as well as acetyl coenzyme A. Acetyl coenzyme A is a 2-carbon atom. It gets converted into 3-carbon atom malonyl coenzyme A. Once after this malonyl coenzyme A conversion, there are 4 steps, that is condensation, reduction, hydration, as well as reduction again. After this whole process will repeat 7 times to form the final product, that is palmitate, which is a 16-carbon chain or 16-carbon fatty acid. And yeah, this is the further process. Once the palmitate is formed, which is saturated fatty acid, it may sometimes get into unsaturated fatty acid in the presence of fatty acyl coenzyme desaturase enzyme, and this happens after the initial fatty acid synthesis. And this is overall overview of fatty acid synthesis. Thank you.
Like, share and subscribe. Hit the bell icon for more updates.